Good morning, students. So this is the 10th class biology first unit nutrition, and we are going to discuss about lab activity, that is oxygen is liberated during photosynthesis. Oxygen is liberated during photosynthesis. How can we prove the oxygen is liberated during photosynthesis? So here, according to our textbook, here experiment to observe oxygen is evolved during photosynthesis in the presence of light. So what are the materials we need? So we need a beaker, test tube, thistle funnel, and hydrilla twigs, and matchsticks, that is matchbox. These are the materials required. So beaker, test tube, thistle funnel, and hydrilla twigs, and matchbox. These are the materials we need. So now what is the procedure? Arrange the apparatus as shown in the figure 5 and make two identical sets with water plants like hydrilla. So here, whenever we have to use the hydrilla only to conduct this experiment, hydrilla plant, it is an aquatic plant, then only we will get exact result. So that is the reason we used to conduct this experiment hydrilla twigs only, hydrilla kommalu matrame, hydrilla twigs. Take the whole set under a bucket of water and invert a water filled test tube over the stem of funnel. So this will help to retain the water column in the test tube. So third one, take the setup out of the bucket and keep it under sunlight. Arrange the second set in the same, cover it with a black paper cloth, keep them under shade. So according to this procedure, we have to take two setup like this. One setup we have to keep in the light, one setup keep in the dark. Now we kept this one in the light, this one we kept in the light, so that here we will get exactly what happened, oxygen liberated. So now how can, so let us take here, and after exposing to the light, after few minutes, what happens, there will be a gas bubbles coming out from the twigs into, that is in the test tube. There will be a gas bubbles, we can see that. So these bubbles are coming from the twigs, hydrilla twigs. So you carefully have to lift up after sufficient gathering of water bubbles in the test tube, and lift up while closing its mouth, test tube mouth, with your thumb. So test tube we have to lift up by closing its mouth with our thumb and upright, and while opening the test tube or while removing your thumb from the mouth of the test tube, you have to ignite the matchstick. So what happen, and keep the matchstick in the that is test tube mouth corner. The matchstick will be glow vigorously and brightly. What happen, from the test tube gas bubbles are coming out. These gas bubbles nothing but oxygen. So this oxygen is liberated by the plants while this exposing to the sunlight. So we should conclude that when green plants can should liberate the oxygen in the presence of light. When we kept in the sunlight, so you should see that the place of water there is air that fills in the setup and kept in sun. It is actually a gas that will be collected in the test tube. Observe other setup kept in the dark. So if there differences you have to find out and in which gas is collected. Test the gas in the test tube by incensing a glowing matchstick or incense stick which would burst into flame. This shows the presence of oxygen. So what precautions do you need while removing test tube from the beaker? Discuss with your teacher. So the gas bubbles need not to be leave from the test tube. That type of observation, that type of care is important, so that while lifting up from the test tube from the beaker, you have to close the mouth of the test tube and lift up and make upright, and slowly you open the mouth of the test tube and keep the matchstick in front of the mouth of the test tube, and the matchstick will be glow vigorously. So this indicates that gas bubbles are formed when we kept the center setup sunlight from the hydrolyte wicks. So in this way we should conclude that oxygen liberated during photosynthesis so by these plants. So this is most important examination level. So